So I'm happy to introduce the next speaker, Dr. Nils Landegren from Uppsala University. He has been interested in a number of autoimmune disorders like Addison's disease, and also he's been interested in the sex difference for autoimmune diseases. And today he's going to talk about one specific autoantibodies, transglutaminase. And the floor is yours.
Thank you so much, Pan, for the kind invitation, and also like to express my gratitude to the Crafoord Foundation and the organizing committee for it's a privilege to be part of this beautiful event, and I very much enjoyed these previous lectures. So the work I will present today on transglutaminases as targets in autoimmune disease. This started for us with an unexpected finding and led to this realization that we have a whole variety of autoimmune disorders connected to different members of this protein family, the transglutaminases. So firstly, what is an autoimmune disease? So this is a topic we have been discussing before lunch. So in contrast to the autoinflammatory diseases, these are disorders that develop from a highly specific responses targeting self-components. So from a clinical point of view, it's a highly variable, right? Autoimmunity may affect different organ systems, but the finding from this group of diseases is that we can see that the adaptive immune response targets self-components, and this is something that we also can define at the molecular level in how B and T cells recognize distinct self-molecules, and that's what we call the autoantigens. So how can we, and I should also say that by studying these target molecules, we can learn a great deal about their manifestations and the involved mechanisms. And this is also something that we use for diagnosing these patients by analyzing autoantibodies in clinical routine. So how can we study and identify these autoantibody targets? So we have heard also during these lectures how we can sequence the genomes, identify causative mutations in monogenic diseases, but can we study in a similar way, can we scan the proteome, identify these autoantibody targets? Well, one approach we have found very useful in our work is to take advantage of these vast panels of full-length human proteins. Using microarray technology, we can study this. So this is an enormous, it's a resource, lots of effort have been put into expressing and purifying these thousands of proteins. And then we can easily take advantage of this and study them in parallel and use for identifying novel autoantibody targets. So the procedure is very simple. We have this array, a conventional microscopic glass slide that we incubate with patient or control serum. And if there are autoantibodies present in the sample, they will bind to proteins on the array, and we can detect them using this secondary fluorescence labeled antibody. So we scan the array and we retrieve the signal for each of these proteins, these 20,000 proteins, covering a majority of the protein coding genes in human. And we first started using this approach to study a very severe autoimmune disease that has a monogenic background, autoimmune polyendocrine syndrome, type 1. So this is a disease that is due to a severe defect in the establishment of central immune tolerance. So the T cells, they are formed in the bone marrow, right? But they need to travel to the thymus where they're selected, and they need to be selected to be able to bind to MHC and exert their functions, but there's also this negative selection with the deletion of autoreactive T cells. So they need to be presented for self-antigens in the thymus. And here is where the R gene comes into play. So the R gene unlocks these otherwise tissue-restricted antigens, so they're presented in the thymus, insulin and so forth. And when this fails in these patients, they develop multiple tissue-destructive, tissue-specific autoimmune components. So we were hoping that can we use this very comprehensive approach to study and better understand the full spectrum of autoimmune manifestations in this disease and learn more about the mechanisms also of thymic tolerance. And what we found that we could replicate and we could confirm these known targets in these different known target organs, the islet cells, we detect GAD antibodies and so forth. But we also made this unexpected discovery of a protein, this strong response in a subset of the patients against a protein that is specific for the prostate, transglutaminase 4. And this first didn't make sense to us. There was nothing described about the prostate being affected in this disease. But what we directly could check in this data was to divide the cohort into male and female patients. And then we could see that at least this very much made sense. So all the positive patients, they were male. So transglutaminase 4, this is a protein that has been studied and known to be crucial. Firstly, it is expressed only in the prostate. So it's expressed in the secretory epithelium of the prostate, and it's secreted to semen, where it carries out important functions. So there are studies indicating this is important for sperm maturation. And what we for sure know is that the Qiang knockout mouse becomes male infertile. And this was interesting for us, given that these patients are often infertile, both females and males with APS1. And female infertility was already explained by the fact that these patients often develop an autoimmune ovarian failure. But the mechanisms underlying male infertility had remained unknown. So this was something we wanted to follow up on and understand better. So firstly, we could confirm very much that this is a bona fide autoantigen. So we replicated this with another method in a larger group of patients, and also where we could confirm that these autoantibodies are present in around half of the males, and they're specific for APS1. But we could take this one step further, given that the prostate, it is formed during embryonic development, but it's first during puberty in the presence of testosterone that it fully matures to an active secretory organ. And that is when the expression of these prostate-specific molecules, PSA and Qiang begins. So looking at these males with APS1, we could see that it was only the males of the adult age, post-pubertal age, that were positive. But there were not that many young males. So as a next step, we could identify these males that were positive for Qiang autoantibodies at adult age, and then go back to the freezers and pick out all the samples that have been collected over the years from these patients at routine visits when they meet their doctors, going all the way back to prepubertal age and early child's age. And when we analyzed these samples for the presence of Qiang autoantibodies, we could see that all these patients had developed this immune response against Qiang sometime following puberty. So it appeared like these individuals were prone to develop this immune response from early in life. But it was first following the expression in the periphery of this protein, when the prostate is maturing, that this autoimmune response was ignited. But how could we take this further and better understand involved mechanisms? We couldn't do much more in this cohort of patients and analyzing these serum samples. But what was possible here in collaboration with Mark Anderson at UCSF was to follow up on this finding in the mouse model of APS1. So the air-deficient mouse, similar to the APS1 patients, spontaneously develops autoimmune manifestations in a number of organs. And interestingly, these mice, they spontaneously develop a tissue-destructive autoimmune prostatitis. And what we could see, that this process was accompanied by the development of Qiang autoantibodies. So this was completely what happened in the mouse, that they spontaneously develop autoantibodies against the same protein as in the patients we studied. And here we could also see that these mice, they become male infertile, and we could also see that the expression of Qiang in the prostate that we know is necessary for male fertility in mouse, this failed completely in the air-deficient mice with the tissue-destructive prostatitis. And we could also see, investigating the thymi of these mice, we could see that Qiang is presented under air control in the thymus. So combining these studies in the patients and the mice very much told us that this could be a mechanism, prostate autoimmunity, contributing to the low fertility in male APS1 patients. So that was the first thing that was interesting for us with this finding of TGN4. But the other thing that interested was that TGN4 is a member of this protein family, 9 members of transglutaminases that have very specialized functions in different tissues and all connected to their abilities to perform a variety of post-translational modification. And importantly, they carry out these protein modifications where they covalently couple proteins to each other or cross-link proteins. And what we were intrigued to learn is that APS1 is not the first autoimmune disease, but instead this is just one of many existing examples. The most known, of course, being celiac disease, characterized by autoantibodies targeting tissue transglutaminase, TGM2. So that is, of course, something that we use routinely for diagnosing these patients. But we also have a subset of patients with gluten sensitivity that develop a skin disease known as dermatitis herpetiformis. And in this case, autoantibodies, they primarily target TGN3 that is expressed in the skin. And we also learned, this was something that was new to me, that a very small proportion of patients with gluten sensitivity, they actually develop neurological symptoms that may involve gluten ataxia. And these patients, they display autoantibodies targeting TGM6 that is expressed in the nervous system. And we also have this acquired form of hemophilia explained by autoantibodies neutralizing factor 13. So this is the coagulation factor responsible for the final stage of the coagulation cascade where fibrin is cross-linked, and this is carried out by a transglutaminase. So we had all these different examples of very clinically distinct autoimmune disorders, but all involving autoimmunity against different members of the transglutaminase protein family. So this, so we asked ourselves, is this the true? Is this the complete list, or can there be even more such examples that remain to be identified? So can we fill out these remaining blanks, and how can this be done? And we had to start someplace, so we started from the top of the list, TGN1, and so now we have this very new problem. So typically, of course, you focus on a particular disease and we try to identify what is the autoimmune target here. But here we had the opposite problem. So we had the candidate autoantigen, but where can we find this disease? But we had some clues. So TGN1, firstly, it's known to be expressed in skin and also other squamous epithelia. And it has these important functions for the stability of epidermis. So if you have biallelic loss of function mutations in TGN1, you are born with a very severe skin disease, a form of ichthyosis. So we were primarily looking for, is there an acquired skin disease that involves autoimmunity against TGN1? And where can we identify it? So then a collaboration with Professor Takashi Hashimoto in Osaka really made this possible for us to pursue this question, since he had this vast biobank with patients with different forms of autoimmune blistering skin diseases. So these patients, they undergo routine diagnostic assessment where you analyze autoantibodies targeting the skin. And this is the traditional method of doing this is using tissue section, tissue substrates, where you stain using indirect immunofluorescence technique. So in these different groups of disorders, you have autoantibodies targeting different components of the skin. So the pemphigus group of diseases, where you have intraepithelial blistering, you find autoantibodies targeting the intercellular junctions in the epidermis, whereas the bullous pemphigoid, or the pemphigo, I should say, group of diseases, you instead find these autoantibodies targeting the basal lamina. But this was the ideal place for us to be looking for this disease, since the only thing that was connecting all these samples that he had collected was that they harbored autoantibodies targeting different components of the skin. In some cases, the molecular targets were known, in some cases not, but we could broadly explore, is TG1 involved in any of the diseases involved collected in his biobank. And in this first assessment of the first samples he sent us, there was this very strong response in a subset of patients with paraneoplastic pemphigus. So this is a very severe blistering disease that involves both the skin and the mucous membranes. And interestingly, this is triggered by an underlying neoplasia. So in most cases, this is a lymphoma, but also other forms of both benign and malignant diseases have been reported. And so we, given these first results, we wanted to confirm this in a larger group of patients. So we retrieved from him, Hashimoto was able to collect all patients that he had reviewed during the last 20 years. And this was another around 90 patients with paraneoplastic pemphigus. And here we could very much confirm that this was a highly specific autoantigen for this group of diseases. So we find these autoantibodies in just more than half of the patients, and it appears to be 100% specific for this particular form of autoimmune blistering disease. So this is something that we hope can aid the diagnostics of these patients, since many of these patients, they first present with these blistering manifestations, and this is how they come in contact with the healthcare. And some of them have this undiagnosed malignancies. So being able to analyze these autoantibodies, detecting Qiang antibodies in a patient with blistering disease, this should, of course, be followed by a thorough investigation to identify potential occult tumors in this patient. So we can for sure add this next example in this even long list of autoimmune disorders, and now there are only 3 remaining ones. So I encourage you all to help us out with this, and maybe we can complete the list together. So we haven't been able to figure these remaining 3 out, but before I end, I would like to share some results that we are really puzzled about, and this is from another transglutaminase, namely TGM3. And this is a protein that is already been implicated with dermatitis herpetiformis. And we studied this disease mostly like a positive control, or for have a complete understanding of the roles of these disorders. And firstly, we could very much confirm that we find these autoantibodies in a cohort of dermatitis herpetiformis. So here we see results from a radioligand binding assay measuring TGM3 autoantibodies, and these are duplicate values, so for 1 to 30 for 31 patients. And in around half of the patients, they are positive for these autoantibodies. But the only problem is that when we also investigate the healthy controls, or the blood donors, I should say, we see this. So we see in 3 of these blood donors, we see a strong response against TGM3. And that is highly reproducible, and that we now also have confirmed with another method. So now we couple TGM3 to beads and use this independent method, where we see a perfect agreement with the result we have obtained. So we can just confirm here that a number of cases, or what we have been doing so far, is to analyze a few hundreds of individuals from different cohorts, and can see that these autoantibodies, they are present in around 1 out of 10 individuals. But we can see that they're slightly increased with age, but we haven't been able to identify or being able to assess any corresponding clinical phenotypes in the cohorts we have analyzed so far. So again, looking at what are the clues here, what could be a corresponding phenotype. Again, the expression pattern is very similar to TGM1 and 5, expressed in the squamous epithelia and important for skin. But what happens if you have mutations in TGM3? Well, the phenotype is very mild, probably due to the overlapping functions in skin between TGM3 and TGM1 and 5. But TGM3 is particularly important for the hair follicles and the formation of the hair shaft. So you develop this uncombable hair syndrome. So these cute kids with uncombable hair may be due to mutations in TGM3. So at this point, we can only say that these around 1 out of 10 individuals walking the streets have these autoantibodies against TGM3. who are these individuals? And at this point, we can only speculate regarding their phenotype. All right. Oh. So with that, I would like to I would particularly like to thank Olle for these studies. So Olle was my PhD advisor, and it's been a privilege to work with him in so many years. And so all these studies were conducted in his lab. And also special thanks to Maribel Arianda-Guyan, who did these important studies on both Qiang 1 and Qiang 3. And also thanks, of course, to Takashi Hashimoto for the studies on blistering disease and Mark Anderson for air-deficient mice. And also thanks for I'm also very grateful for collaboration not presented here, but with Van Hammarström and Jean-Laurent Casanova. So with that, I thank you so much.
Yeah.